Hello and welcome back to Bobo's Biology channel, where we explore the molecular, cellular and systemic mechanisms that govern life with rigorous evidence-based scientific analysis. Drug development is one of the most scientifically rigorous and high-stake endeavors in biomedical science. While many drugs successfully make it to the market, a surprisingly large number fail at crucial stages. Understanding these failures and the lessons learned from them is critical for students in medicine, pharmacology, biotechnology, and related disciplines. Today, we will explore several high-profile drug failures, each illustrating a different mechanism of failure. First, we will talk about TGN-1412, a catastrophic Phase 1 safety failure. Then we'll talk about Alzheimer's drugs and repeated Phase 2 and Phase 3 efficiency failures, followed by Vioxx, a post-marketing safety withdrawal. Afterwards, we'll talk about SPK-9001, Phase 1 and Phase 2 clinical setbacks in hemophilia B gene therapy. Then about vaccines and public perception and the societal impact on drug success. And finally, the commercial failures despite regulatory approval. For each, we will explore what happened, why it happened, and the key scientific or regulatory lessons. In March of 2006, a first-in-human trial in London of TGN-1412, a humanized IgG4 monoclonal antibody targeting CD28, triggered life-threatening reactions in all 6 volunteers within hours. Designed as a superantagonist, TGN-1412 could activate T cells via CD28 alone, bypassing the normal antigen TCR signal. The therapeutic goal was to expand regulatory T cells, also called Tregs, to suppress autoimmunity. So what went wrong? Preclinical studies in macaques showed only Tregs and naive CD4 plus T cells responding to TGN-1412. In turn, Tregs proliferated and then were able to secrete immunosuppressive molecules like IL-10 and TGF-beta. CD4 plus naive T cells proliferated too and then secreted very little IL-2, an inflammatory cytokine, to support proliferation. Regulatory T cells, however, possess a special receptor called CD25, which shows very high affinity for IL-2. So that caused the overall response in preclinical studies to be immunosuppressive. However, in the Phase 1 first-in-human trial, something else happened. Memory CD4 plus T cells and effector CD4 plus T cells were activated and instantly released big amounts of inflammatory cytokines, mainly TNF-alpha, IFN-gamma, IL-2 and IL-6 before proliferating, thereby responding very fast. Tregs, on the other hand, were activated, however their cytokine production after activation is slower since they first need to proliferate before producing immunosuppressive cytokines. Thereby, and by the clustering of CD28 molecules amplifying the signal, a massive cytokine production, also called a cytokine storm, was triggered. The lessons we can learn from this are that the human immune response can diverge substantially from those observed in preclinical animal models. For example, in macaque studies of TGN-1412, memory and effector CD4 plus T cells lacked surface expression of CD28 receptors, preventing engagement by the drug, whereas human CD4 plus T cells expressed CD28 and responded robustly. This discrepancy contributed to the severe cytokine release syndrome observed during the first-in-human administration. Current protocols of first-in-human studies of immune-modulating biologics incorporate staggered and dose-escalated administration, comprehensive in vitro profiling of human immune cell responses, and enhanced real-time clinical monitoring, specifically to mitigate the risk of cytokine storms and other unexpected systemic immune reactions. Alzheimer's disease presents one of the most challenging targets in modern medicine. Despite decades of investments and biological insight, many promising candidates have failed late in clinical development. For decades, the amyloid hypothesis has dominated Alzheimer's research. The idea, accumulation of amyloid beta or A-beta in the form of plaque in the brain drives neurodegeneration. Many drugs have been developed to reduce amyloid levels, including beta secretase inhibitors, gamma secretase inhibitors, and anti-amyloid monoclonal antibodies. Let's first understand how A-beta is normally produced, by looking at the non-amyloidogenic and amyloidogenic pathways side by side. In the non-amyloidogenic pathway, alpha secretase cleaves amyloid precursor protein within the amyloid beta sequence. In parallel, the amyloidogenic pathway begins with beta secretase cleaving amyloid precursor protein at the N-terminal of the amyloid beta sequence. Thereby, the non-amyloidogenic pathway produces after the step the soluble fragment SAPP-alpha and a membrane-bound C83 fragment. In the amyloidogenic pathway, SAAP-beta and a membrane-bound C99 fragment are being produced by this step. In the following step, in both the non-amyloidogenic and amyloidogenic pathway, gamma secretase cleaves the intracellular portion of the respected protein. In the non-amyloidogenic pathway, gamma secretase is cleaving C83 to release a small peptide called P3, which is non-toxic, and the APP intracellular domain called AICD into the cytoplasm. In the amyloidogenic pathway, gamma secretase cleaves C99, releasing amyloid beta 40 peptides into the extracellular space. Amyloid beta 40 is produced naturally in the brain and in its normal form is non-toxic. However, if something changes either in how gamma secretase recognizes its cutting site or in the sequence of the amyloid precursor protein itself, the enzyme can cut a slightly different position. This produces a longer isoform called amyloid beta 42. Amyloid beta 42 is more hydrophobic, meaning it sticks to itself more easily. As a result, it aggregates into toxic oligomers and plaques, disrupting synaptic function, damaging neurons, and ultimately contributing to Alzheimer's disease. Now let's see how drugs intervene here. Beta secretase inhibitors target the very first step of the amyloidogenic pathway by blocking beta secretase. C99 is therefore not produced, and therefore no amyloid beta 40 nor amyloid beta 42 is formed. This reduces amyloid load upstream before toxic peptides even exist. Gamma secretase inhibitors act downstream, blocking the enzyme that releases amyloid beta from C99. Interestingly, gamma secretase is also responsible for cleaving C83 in the non-amyloidogenic pathway, which produces P3. Blocking gamma secretase therefore affects both pathways. However, P3 is considered non-toxic and its absence does not appear to have major detrimental effects, unlike amyloid beta 42 accumulation, which is pathogenic. This is why gamma secretase inhibition was initially pursued despite potential pathway overlap, though off-target effects on the other substrates, like Notch, remain a challenge in therapy development. Finally, anti-amyloid monoclonal antibodies work differently. They target extracellular amyloid beta peptides or plaque, binding to them directly. This marks plaque for clearance by microglia, reduced soluble oligomers, and can partially dissolve aggregated deposits, ultimately lowering neurotoxicity. Importantly, these antibodies do not affect antigen precursor protein processing, so production of new amyloid beta continues unless combined with secretase inhibitors. In summary, understanding the balance between alpha and beta secretase processing and targeting these pathways strategically remains central to reducing amyloid burden in Alzheimer's disease. Therapeutics can either prevent formation upstream or clearing existent amyloid beta, offering complementary strategies to slow or halt disease progression. Why these drugs failed? Despite reducing amyloid burden in some cases, most agents showed no meaningful cognitive improvement despite the reduction of amyloid plaque burden. Most anti-amyloid therapies have failed to produce statistically or clinically significant gains in standardized cognitive assessments. No slowing of functional decline. Even with biomarker evidence of target engagement, patients generally do not show measurable preservation of daily living activities or functional independence, highlighting the gap between amyloid clearance and meaningful clinical benefit. Potential for worsening outcomes or adverse effects. Some trials reported cognitive worsening, brain edema, microhemorrhages or ARIA, amyloid-related imaging abnormalities, illustrating that amyloid-targeting therapies can carry serious safety risks, particularly when administered to patients with advanced neurodegeneration. The key scientific issues are target validity. The causal role of amyloid accumulation in Alzheimer's disease progression remains unsolved, and amyloid deposition may represent a downstream or epiphenomenal process rather than the primary pathogenic driver. Disease progression window. Clinical trials often target patients at symptomatic stages following prolonged amyloid accumulation and downstream neurodegenerative cascades. At this point, widespread neuronal and synaptic degeneration has already occurred, reducing the likelihood that amyloid-targeting therapies can alter disease trajectory. And finally, inadequate biomarkers. Reliance on surrogate biomarkers, such as amyloid PET burden, has not consistently correlated with meaningful clinical or cognitive outcomes. Cognitive decline reflects complex multifactorial processes that are not fully captured by amyloid-centric biomarkers alone. Several drug candidates have made it to phase 3, only to fail at efficiency endpoints, costing billions in research investments. The lessons learned from that are that disease mechanisms must be validated using clinically meaningful endpoints rather than relying solely on surrogate biomarkers that may not capture downstream neurodegenerative processes. Second, therapeutic intervention may need to occur at prodromal or preclinical stages prior to extensive synaptic and neuronal network disruption to meaningfully alter disease trajectory. And lastly, multifactorial neurodegenerative disorders are unlikely to respond robustly to single-target interventions, necessitating combination or system-level therapeutic strategies. In 1999, Vioxx was approved as a COX-2 selective non-steroidal anti-inflammatory drug called NSAID. It was intended to relieve pain while sparing gastrointestinal side effects seen in many traditional NSAIDs. Vioxx acted on the arachidonic acid cyclooxygenase pathway. This pathway is the central biochemical route for prostanoid synthesis. It begins at the cell membrane, where inflammatory or mechanical stimuli activate phospholipase A2, releasing arachidonic acid from the membrane phospholipids. Arachidonic acid is then metabolized by cyclooxygenase enzymes COX-1 or COX-2, which catalyze its conversion to prostaglandin H2 or PGH2. PGH2 is a common unstable intermediate that serves as the branching point for multiple biological active prostanoids. In the gastrointestinal tract, constitutively expressed COX-1 converts prostaglandin H2 into protective prostanoids that maintain gastric mucosal blood flow, stimulate mucus and bicarbonate secretion, and preserve epithelial integrity, making preservation of this pathway a central therapeutic goal. In platelets, prostaglandin H2 is converted into thrombohexane A2, a potent vasoconstrictor and promoter of platelet aggregation that supports hemostasis and thrombosis. In endothelial cells, prostaglandin H2 is converted into prostacyclin, which induces vasodilation and inhibits platelet aggregation, thereby promoting vascular protection. In inflamed tissues, inducible COX-2 converts prostaglandin H2 into prostaglandin E2, a key mediator of pain, fever, and inflammation. Selective COX-2 inhibition was therefore designed to suppress inflammatory prostaglandin E2 production while sparing COX-1-dependent gastric protection and platelet function. In clinical trials, COX-2 inhibition efficiently reduced prostaglandin E2-mediated inflammation and pain while sparing COX-1 activity. As a result, gastric prostaglandin synthesis and platelet function were largely preserved, leading to improved gastrointestinal safety compared to non-selective NSAIDs. However, COX-2 also contributes to endothelial prostacyclin production. After Vioxx entered widespread clinical use, post-marketing epidemiological studies revealed that chronic COX-2 inhibition reduced prostacyclin levels while leaving platelet-driven thrombohexane signaling intact. This imbalance shifted vascular signaling towards vasoconstriction and thrombosis, increasing the risk of myocardial infarction and stroke, particularly with long-term exposure in patients with underlying cardiovascular risk. These adverse effects were not detected in pre-approval trials due to limited sample size, shorter duration, and exclusion of high-risk populations. Only after millions of patients were exposed did the cardiovascular safety signal become apparent, ultimately leading to the withdrawal of Vioxx in 2004. The lessons learned from that are that pre-approval clinical trials are inherently limited in their ability to detect rare, delayed, or population-specific adverse effects due to constraints in sample size, trial duration, and patient selection. Another lesson is that post-marketing pharmacovigilance is critical for identifying low-frequency and long-latency safety signals that only emerge under conditions of widespread and prolonged real-world exposure. The last and most important lesson learned is that therapeutic risk-benefit assessments are dynamic and must be continuously re-evaluated as new safety and efficiency data accumulates across diverse patient populations over time. In the 2010s, Sparks Therapeutics developed SPK-9001, an adeno-associated virus-based gene therapy intended to treat hemophilia B. Hemophilia B is a rare inherited bleeding disorder caused by mutations in the F9 gene, which encodes the coagulation factor 9. The disease is X-linked and primarily affects males. Individuals with hemophilia B have reduced or absent functional factor 9, leading to impaired blood clotting. Clinically, this results in prolonged bleeding, spontaneous hemorrhages, and excessive bleeding following trauma or surgery. Standard treatment for hemophilia B involves regular intravenous infusions of recombinant or plasma-derived factor 9 to prevent bleeding episodes. While effective, this approach is burdensome, expensive, and does not provide a permanent cure. In response to these limitations, Sparks Therapeutics developed SPK-9001, an adeno-associated virus-based gene therapy designed to provide long-term correction of the underlying genetic defect rather than temporal replacement of the missing protein. When a blood vessel is injured, the endothelial lining is disrupted, exposing subendothelial collagen fibers and negatively charged surfaces. This exposure initiates the intrinsic coagulation pathway. Circulating factor 12 comes into contact with exposed collagen and is activated to factor 12A. Factor 12A then activates factor 11, converting it to factor 11A. Activated factor 11A cleaves and activates factor 9, converting it to factor 9A, which represents a critical control point in the intrinsic pathway. Once activated, factor 9A associates with its cofactor, factor 8A, along with calcium ions, on the phospholipid surface of activated platelets. Together, these components form the intrinsic tenase complex. This complex catalyzes the conversion of factor 10 into factor 10A, a key amplification step in the coagulation cascade. Factor 10A subsequently drives thrombin generation, and thrombin converts soluble fibrinogen into insoluble fibrin. Fibrin molecules polymerize and cross-link to form a stable fibrin mesh that seals the wound and prevents further blood loss. However, in hemophilia B, F9 mutations can lead to several distinct defects. In many cases, factor 9 is produced in markedly decreased quantities, resulting in insufficient circulating protein available for activation. In other cases, factor 9 is synthesized and post-translationally modified but contains structural abnormalities that impair its function. Such defective factor 9 may fail to be efficiently activated by factor 11A, or, once activated, may be unable to bind factor 8A, calcium ions, or phospholipid surfaces on activated platelets. These defects prevent proper assembly of the intrinsic tenase complex. As a result, activation of factor 10 is severely impaired, leading to reduced thrombin generation and insufficient conversion of fibrinogen to fibrin. The inability to form a stable fibrin mesh ultimately results in defective clot formation and the bleeding phenotype characteristics of hemophilia B. Gene therapy using SPK9001 was designed to correct this defect by delivering a functional copy of the F9 genes to hepatocytes using an adeno-associated viral vector. The therapeutic strategy aimed to restore endogenous production of properly modified factor IX in the liver, thereby re-establishing normal clotting function and eliminating the need for repeated factor infusion. Although early clinical trials demonstrated increased circulating factor IX levels, some patients developed immune-mediated liver toxicity. Pre-existing immunity to the adeno-associated virus triggered cytotoxic T-cell responses against transduced hepatocytes, causing liver inflammation. This immune-mediated destruction of hepatocytes resulted in the loss of the episomal transgene, since the DNA is confined to the cells and not integrated into the genome. Consequently, factor IX expression declined, limiting the durability of the therapy and highlighting the challenges of achieving long-term efficiency and immune tolerance in liver-directed gene therapy. The lessons learned from this were that preclinical animal models did not adequately predict human immune responses to the adeno-associated viral vectors, underscoring limitations in translational immunogenicity assessments. Pre-existing humoral and cellular immunity to the adeno-associated virus can result in cytotoxic T-cell-mediated elimination of transduced hepatocytes, compromising both safety and durability of gene expression. The episomal persistence of the adeno-associated virus-delivered transgene, while minimizing genomic integration risks, renders therapeutic expression vulnerable to loss following immune-mediated hepatocyte injury. Liver-directed gene therapy requires careful patient selection, including screening for anti-adeno-associated viral antibodies and optimization of vector design, dosing, and immunosuppressive strategies to achieve sustained therapeutic benefits. These findings highlight the necessity of integrating immunological considerations into the development and clinical deployment of viral gene therapies. Vaccines and public perceptions. The societal impact on drug success. Scientific validation alone does not ensure societal adoption. Vaccines undergo rigorous preclinical and clinical testing and continuous post-marketing surveillance, yet public skepticism persists. Key challenges include rapid dissemination of misinformation via social media, amplification of extremely rare adverse events, and distrust in regulatory and health institutions. The impact of these factors includes reduced vaccination coverage, resurgence of preventable diseases, and increased burden on the healthcare system. Mitigation strategies involve transparent, contextualized risk communication, community engagement with trusted stakeholders, and data-driven responses to misinformation. Public perception functions as a parallel socio-biological system that can enhance or undermine biomedical success. Commercial failure despite regulatory approval. Regulatory approval does not guarantee commercial success. Market failures can occur when a drug offers minimal clinical advantage, faces pricing or reimbursement barriers, encounters poor marketing or adoption challenges, or competes with superior alternatives. Real-world impact requires both scientific success and strategic integration into the healthcare system. Across these examples, one central lesson emerges. Drug development is fundamentally multidimensional. Therapeutic success depends not only on the molecular target engagement and clinical efficiency, but also on robust translational safety assessments, populational specific biology, immune and genetic variability, societal acceptance, and strategic integration into real-world healthcare systems. Collectively, these cases demonstrate that setbacks are not simply obstacles, but critical sources of information. Each failure refines mechanistic understandings, exposes limitations in experimental models, improves trial designs, and sharpens risk-benefit evaluations. When systematically analyzed, these outcomes directly inform safer, more effective, and more equitable therapeutic strategies. Ultimately, drug discovery and clinical development are iterative processes driven by evidence, adaptation, and learning. Progress is built not on uninterrupted success, but on the careful interpretation of unexpected results. Each challenge encountered strengthens the scientific foundation of future therapies, ensuring that innovation in medicine moves forward with greater precision, responsibility, and impact.